Good morning, it's June 30th, and this is your daily brief in science. Here's everything you need to know. Researchers at the University of Cambridge have made a significant advancement in cancer treatment with a new test that predicts how patients will respond to chemotherapy by analyzing tumor DNA. This innovative approach has the potential to spare patients from unnecessary treatments by identifying specific DNA signatures and patterns within their cancer. In a pilot study that included 840 patients, the test effectively categorized individuals as either chemotherapy-resistant or chemotherapy-sensitive. This categorization allows doctors to create personalized treatment plans tailored to the unique needs of each patient. Professor James Brenton from the Cancer Research UK Cambridge Institute highlighted the critical issue of chemotherapy resistance, noting the unpleasant side effects that often accompany treatments that yield limited benefits. Chemotherapy has remained largely unchanged for the past 40 years, prompting experts to advocate for more personalized approaches to enhance treatment effectiveness while minimizing adverse effects. Dr. Ian Folks from Cancer Research UK remarked that the era of one-size-fits-all chemotherapy is coming to an end, as personalized treatment strategies become increasingly feasible. The test connects DNA mutation patterns to the mechanisms behind chemotherapy resistance, providing valuable insights that could improve patient outcomes. Designed for user-friendliness, the test integrates well with existing genomic sequencing methods, making it practical for clinical implementation. It specifically targets 3 common chemotherapy types: platinum-based, anthracycline, and taxane, which are known for their toxicity and severe side effects. Results from the study showed that patients predicted to resist certain chemotherapy types, such as anthracycline and platinum, experienced higher treatment failure rates in cancers like ovarian and metastatic breast cancer. This ongoing research emphasizes the importance of tailored treatment strategies, which could lead to longer and healthier lives for cancer patients. The test has been piloted using data from hundreds of patients across various cancers, particularly focusing on prostate, ovarian, and breast cancers. Recent research from the Galápagos archipelago reveals that tomatoes on the harsher western islands are synthesizing alkaloids akin to those produced by their ancient ancestors. This marks a significant departure from modern tomato varieties and introduces a phenomenon termed de-evolution. Researchers suggest that environmental pressures are causing these tomato plants to revert to a more primitive genetic state. The extreme conditions on these islands, characterized by barren landscapes and poor soil, may be driving these plants to adopt older traits that enhance their survival. The ancestral alkaloids produced could provide better defense mechanisms against these harsh environments, prompting this reverse evolution. The study indicates that notable biochemical shifts in these plants can be traced back to modifications in just 4 amino acids within a single enzyme responsible for alkaloid production. These small genetic changes illustrate how even minor alterations can lead to significant transformations in plant biochemistry. Published in Nature Communications, these findings challenge traditional evolutionary views by highlighting the dynamic nature of genetic adaptation in response to environmental changes. The implications extend beyond tomatoes, suggesting that similar factors could lead to the reemergence of ancient traits in other species, including humans. This raises complex questions about evolution, indicating that traits once considered lost may actually be dormant and can reappear under specific conditions. Lead researcher Adam Jozwiak emphasizes that this study could prompt a reevaluation of evolutionary theory, with potential applications in genetic engineering, including the development of new medicines and pest-resistant crops. Supporting this research, similar genetic changes were induced in tobacco plants, which began producing the ancient alkaloids, further underscoring the study's findings. A recent study from the Yunnan Institute for Endemic Disease Control and Prevention has unveiled alarming findings regarding bat-borne viruses. Over 4 years, researchers analyzed the kidneys of 142 bats from 10 different species, identifying 22 viruses, 20 of which were newly discovered. These viruses, found in bats from the Yunnan region of southern China, are linked to severe encephalitis and fatal respiratory diseases. In addition to these viruses, the study also revealed a new protozoan, Klebsiella yunnanensis, and 2 new bacterial species. This suggests that bats may be harboring a greater variety of dangerous pathogens than previously recognized. Notably, the viruses were predominantly found in Rusetus leschanatia fruit bats, which dwell near orchards and residential areas, raising concerns about zoonotic transmission to humans, particularly through contaminated fruits. Professor Vinod Balasubramaniam from Monash University pointed out the increased risk of transmission due to the bats' proximity to human populations. The study emphasizes the urgent need for comprehensive microbial analyses to evaluate the risks associated with bat-to-human transmission of these viruses. Alarmingly, the newly identified viruses show a genetic similarity of 52 to 57% with the deadly Nipah and Hendra viruses, both of which currently have no known cures or vaccines. Yunnan's geographical and climatic conditions are similar to those in Malaysia, where Nipah outbreaks have occurred, suggesting an elevated risk of virus spread. Bats are recognized as natural reservoirs for numerous microorganisms that have historically led to epidemics in humans, including SARS and COVID-19. Previous research has linked bats to the transmission of several deadly diseases, underscoring the necessity for systematic monitoring of bat populations worldwide. The study's innovative approach, examining the internal organs of bats instead of just fecal samples, enhances the likelihood of identifying pathogens that could pose risks to humans and animals. Astronomers have made a groundbreaking discovery by detecting ancient radio signals from the distant galaxy cluster ESP-ARCS 1049. This finding could provide new insights into the formation of the early universe. Utilizing the Low-Frequency Array, or LOFAR, which comprises 100,000 antennas across 8 European countries, the research team identified a faint radio signal from ESP-ARCS 1049. Remarkably, these radio waves have traveled for an astonishing 10 billion years to reach Earth, marking the first detection of such signals at this immense distance. The mini-halo, which spans over 1 million light-years, represents a structure previously only observed in nearby galaxies. These mini-halos consist of faint groups of charged particles that emit both radio and X-ray waves, typically found in clusters between galaxies. The researchers proposed 2 primary explanations for the formation of this mini-halo: the activity of supermassive black holes ejecting high-energy particles, and cosmic particle collisions occurring in the cluster's hot plasma. These findings suggest that black holes or particle collisions may have energized galaxies earlier than previously believed, enhancing our understanding of cosmic evolution. Co-lead author Julie Hlavaček-Larondo expressed optimism that this discovery opens new avenues for studying the energetic conditions of the early universe. Meanwhile, co-author Roland Timmerman emphasized the significance of the strong radio signal, indicating that energetic particles have been shaping galaxy clusters for nearly the entire history of the universe. Future telescopes, such as the Square Kilometre Array, will be essential in further investigating these phenomena and understanding the influence of magnetic fields and cosmic rays on the evolution of the universe. This research, accepted for publication in the astrophysical journal Letters, was co-led by Hlavaček-Larondo from Université de Montréal and Timmerman from Durham University's Institute for Computational Cosmology. Overall, this discovery provides valuable insights into the nature of galaxy clusters shortly after their formation. Researchers at the University of Pennsylvania have made a groundbreaking discovery that could change the landscape of leukemia treatment. They have identified a compound derived from the fungus Aspergillus flavus, historically known for its toxicity, which now shows promise as a potential therapeutic opportunity. Led by Kuyue Ni, the research team focused on specific proteins and genes in Aspergillus flavus that are responsible for synthesizing ribosomally synthesized and post-translationally modified peptides, or RIPPs. This investigation led to the identification of 4 unique compounds called asparagimycins, which have demonstrated the ability to inhibit cancer cell growth by blocking microtubule formation, a critical process for cell division. In laboratory experiments, 2 variants of the asparagimycins exhibited strong effects against leukemia cells, with one variant showing efficacy comparable to established FDA-approved treatments such as cytarabine and daunorubicin. To enhance their effectiveness, researchers modified these compounds to improve their penetration into leukemia cells, utilizing specific transporters to increase therapeutic potential. The study suggests that asparagimycins could disrupt cancer cell division, indicating a targeted approach in leukemia treatment, while showing minimal effects on other cancer types and microbes. However, it is important to note that the compound displayed limited effects on breast, liver, and lung cancer cells, highlighting its specificity. Despite these promising results, caution is warranted as the research is still in its early stages, conducted on cell cultures. Further studies are needed to evaluate the compound's efficacy in human subjects. Lead researcher Sherry Gao emphasized the significance of Aspergillus flavus, which, despite its historical association with respiratory issues and fatalities, harbors unexplored gene clusters that could yield beneficial compounds, reminiscent of the historical development of penicillin. This has been your daily brief in science. To read more about these stories, follow the links in the episode bio. You can also subscribe to these updates via email at www.brief.news. For more daily podcasts about the topics you love, visit brief.news/podcasts. Tune in tomorrow. We'll be back with everything you need to know.